Hi, so welcome back to chapter 4, not back to chapter 4, welcome back to microbiology and welcome to chapter 4 for the first time. Well, unless you're rewatching this, I guess. Yeah, this is going to be talking about the prokaryotes. If you guys don't remember, a prokaryote, pro being before, and karyote, karyo means like nucleus. So before nucleuses existed. So then we're talking about bacteria and archaea. Remember, because we are eukaryotes and we would have had to have come after that. So, bam. Okay, what are we going to learn in this chapter? The structures of bacteria, the shapes and arrangements of bacteria, the components of bacterial flagella and how it propels bacteria around, how they move with it, the difference between the two types of bacterial envelope structures. That's going to be talking about the gram-positive and the gram-negative. Those are the major ones that you're going to need to worry about. There's another one called acid-fast that you will have to know about as well. The mechanisms of sporulation and germination, differences between archaea and bacteria, and the 4 divisions of bacteria and their characteristics. I mean, like I said, it's one of my favorite stuff. So these are the parts of bacteria, a little bit on each one because it's super fun talking about it. All bacteria have a cell membrane of some kind. They have at least one cell membrane on the inside that contains their cytoplasm, which is the liquidy stuff on the inside, and whatever components elsewise that could be in there. They also have a chromosome or a nucleoid. That's usually a nucleoid region. That means that's where their DNA and stuff are located. Since they do not have a nucleus, they just have a region where that stuff is chilling. Then they have ribosomes. Every single one of them has a ribosome. Those are the things that help make protein, okay? They synthesize protein, the protein manufacturers. And then we have the cytoplasm itself, which is the little watery stuff on the inside. So all bacteria have those 4 things, okay? Some bacteria also have an S layer, which is a layer used for attachment and stuff like that. Fimbriae, which is also used for adhesion and attachment. Outer membrane, that's usually like for gram-negatives. Gram-negatives have an inner membrane and like a thin wall and then an outer membrane, okay? So some of them have an outer membrane. Some of them have a cell wall. There are some that do not have cell walls at all, like mycoplasma, don't worry about it, but some of them don't have a cell wall. What is a cell wall for bacteria? Don't worry, we'll talk about it, but it's made of peptidoglycan, okay? The actin cytoskeleton, that's just literally what that keeps the cell shape. Okay, the pilus, it's like the sex pilus, also is what they call it. It's how they transfer DNA between one another. So if they have like a plasmid, which is an extra little bit of DNA that they can carry special genes on for like self-protection or whatever, like antibiotic resistance, they can push it through that to other ones, to other bacteria that are like nearby. Okay. The capsule, which is like a kind of sticky coating that allows for adhesion, avoiding the immune system and stuff like that. And also includes like the slime layer and also that stuff. So the inclusion vacuole or like granules or stuff like that, they're just a junky thing in the middle. It contains stuff. Don't worry about it. It's just junk in there. Bacterial microcompartments, again, similar thing, just junk. Don't worry about that. It contains whatever stuff. The plasmid, that's the extra little bit of DNA. It's not part of the actual chromosome itself, but it does confer things like antibiotic resistance, extra genes that help it out. And then the flagellum, some of them have, that is for locomotion, moving around. Right, some bacteria also have, we don't have it in the picture, but they also have endospores, which we talked about last time, which helps them survive harsh conditions. They are not metabolically active, the endospores, but they are waiting for better conditions so that they can continue to, multiply again or whatever in the vegetative state. Then this is the other thing that they can do, which is the intracellular membrane. I mean, I don't know. We're not going to talk about that one. So, bacteria and archaea versus eukaryotes, okay? Bacteria and archaea do not have a true nucleus. They do not have membrane-bound organelles at all. So what do they do with their DNA? Bacteria and archaea have their nuclear material, their genomes and stuff, just free in the cytoplasm, whereas we, eukaryotes, have them in a nucleus. Cell walls. The cell walls of bacteria is made of peptidoglycan. That's an important term. Be familiar with it. Be able to at least at the very least, recognize this and know what it is. It is the molecule that makes up bacterial cell walls. Archaea have cell walls, but it's different from both of them. That's all you really need to know. And then eukaryotes, they have cell walls, but eukaryotes that have cell walls, the fungi, for example, they have chitin-based cell walls. And then plants have cellulose-based cell walls, okay? Then they have the internal structures, whereas bacteria and archaea don't have membrane-bound organelles. Eukaryotes have several. They have a lot, like mitochondria, endoplasmic reticulum, the Golgi apparatus, all of these. So bacteria are single-celled organisms. They're capable of carrying out all of life activities. Each of the cells are individually. They can reproduce, carry out metabolism, activities, and then process nutrients. They can also work as a group, either when they're living in a colony or in like a biofilm. They have like ways to signal each other in the biofilm to tell each other how things are going and what's going on. When bacteria are growing, depending on how they divide, whether they like divide and then like create little chains next to each other, or divide and they kind of grape cluster up, or whatever's going on, that is what's referring to their bacterial arrangements. And their size and stuff like that. We talked about that previously. Like they have a lot of variety in the size. And we have the actual cell shapes themselves. Those can vary too. So anyways, moving on, we talked about the size. I'm not as concerned. I'm not ever going to ask you those numbers. The three general shapes of bacteria. The cocci is the spheres, or somewhat oval shaped or bean shaped. The bacillus, they are cylindrical, like rods. Okay, that's what the bacillus are. They're rods. And then spirillum are helix shaped or squiggle shaped. Okay. Pleomorphism is a term that refers to variations in size and shape among cells of a single species. So if that single species sometimes has a lot of variety in its shape, then we call it pleomorphic. Okay. Pleo being many, morph being shape. All right, so this is pictures of the cocci in the far left. The middle is the bacillus. It's different variations that you might see in a bacillus, and then vibrio, which is like a comma shape or whatever. So that's the idea of what you might see in the shapes. And here's some spirillum on the far left, the spirochetes, which are like tighter wound, and then branching filaments here on the end on the right. So those are like arrangements, okay? So going back to the shapes and the groupings and all of this, the diplococci, that's a word. So we're talking about the cocci. That's a word that means you have two cocci, right? Diplo, di, like that's two. Don't argue with me on that one, right? So diplococci mean you have two cocci together. So what would that be? That would be this one here. If you want just a single cocci, that is this one here. You can have tetrads, which is 4. So that's this one here. Then we have the staphylococci, which is the irregular clusters. It's not very irregular looking here, but this is the idea. They can be sort of different shapes like that. The streptococci, I think of strips. So streptococci almost, but they're streptococci in strips or chains, okay? Strips or chains, they're linked end to end. And then the sarcina, which is a cubital packet, okay? So those are the different arrangements and stuff. So this is like, so diplo, tetrad, staphylo, strepto, and sarcina. These can apply not just to the cocci, but maybe to the bacilli as well. It doesn't have to be just, so this is the arrangement more than it is the shape. So diplobacilli, and moving on to the bacilli, that's a pair of bacilli. So we have the monobacilli, the coccobacillus, which is like a combination of both to get you real confused. Diplobacilli, 2 of them. Palisades, they kind of are, they were made like end to end, so they're still stuck together on their little ends, however they divide it out. And that looks like a picket fence. So palisade is like, they had like fences up for protection. I mean, I don't know why. There's this song that I heard about palisades. Now that's all I think about every time I see that. Anyways, moving on. Strep, right? We talked about this. So spirilla are occasionally found in short chains, but usually not. And spirochetes that are very tightly wound, they rarely remain attached after cell division. So they're not usually, they're just going to be by themselves, like individuals. So what are the arrangements of the bacteria pictured here? We have some cocci, but that's not the arrangement, that's the cell shape. Be very clear about cocci, bacilli, what was the other one? Spirillum, spirochetes, that's the shape of the cell. The arrangement is talking about staphylo or strepto or palisade, right? So this looks to me like strepto. So I would go with D, that's strep, it's like strepto, right? Chains. Okay, external structures. We're talking about the flagella and the axial filaments. So those are for movement, okay? We also talked about the fimbriae and the pili, that's for adhesion. And the channels, like we said, transferring the sex pili. So this is the flagella. This is electron microscopy of the flagellum and what it looks like, that motor or whatever. These are literally made of proteins. This is not like a mechanical thing, right? This is actually just made of proteins. This is organic molecules that we talked about before, and just how they're sticking together. And that arrangement is what makes up this flagellum. It is not going to whip about like this. It's going to rotate. Okay, and that's what's going to allow for the movement. So you can have polar flagella. So that means that you have flagella at both ends or one end. So not all over the place, either one or both ends. If you are monotrichous, monotrichous means a single flagellum. Lophotrichous, I don't know why that sounds like puffy to me. So I always think small bunches or tufts. It sounds like it should be tufts. That word is terrible. I'm sorry for that drawing. Amphitrichous, we had talked about amphi before, right? That means both. Remember, because we had talked about amphipathic molecules like phospholipids that had the polar head and the hydrophobic tails. Well, this is just like this, amphitrichous. So we're talking about amphi, just think trichous, just think of flagella. Don't worry about what it actually means. So if it's amphitrichous, we have both. So it's both ends of the cell. So amphitrichous at both ends. And then we have peritrichous. Peri is just referring to all over. So it's just covered with flagella. So what type of flagellar arrangement has appendages at both poles of a rod-shaped cell? That we just said, both, that's amphitrichous. Okay, moving on, the function of flagella. Why would we need to move around? So one of the reasons could be chemotaxis. What does that mean? It's movement in response to chemical signals. We can have positive chemical signals, so positive chemotaxis. We're moving towards something that we want. Negative chemotaxis is moving away from something that we don't want, like a harmful thing. Okay, so positive, yeah, move towards it. Sounds good to me. Negative, I don't want to do it. That's the idea. Phototaxis is movement towards light. It's another thing we can do. When we use our flagella, we can move in runs and we can move in tumbles. If we are moving in runs, we will have a counterclockwise movement of our flagella, and the cell will swim in a smooth linear direction towards whatever that stimulus is, or away from the stimulus, whatever direction you're trying to go, right? The tumble is a flagellum where the flagellum reverses its direction suddenly. It'll cause the cell to stop suddenly and change its course just randomly. And if you have a repellent that suddenly appears, then that's often what causes numerous tumbles. So it's just trying to get away however it can, because it doesn't necessarily know which direction to go. It has to sense its environment around itself in order to know how to keep moving. So, and how would it do that? Well, it's just like chemical signals that are on the outside that it would have to respond to. So this is the idea with the runs and the tumbles, okay? So if we had one flagellum, this is what it would look like versus if you were like peritrichous, which is just covered in flagella. But yeah, you've got to run, and then the tumble, you stop, and then you jerk, and then you start going again, and you jerk again, and then whatever. So that's a tumble. This is movement of the cell using the flagellum. We have the tumbles and the runs. The tumble is essentially a stop almost because it's a sudden change of direction. So that's in the red, and then the runs are in green. So we can see here that if we have no attractant or repellent and we have a random movement, we see that here in the figure A, but in the presence of an attractant where it wants to go somewhere over here, we can see a generalized movement and less tumbling randomly and more just overall movement in a direction. It's not so super perfect, but it gets there. There's another kind of flagella, the periplasmic flagella, which is on the inside. It's an axial filament, which is literally just like a filament, like a thing that's on the inside. Okay. Essentially, they're coiled threads inside of the cell. They're an internal flagellum. And why would you want that? That allows for twisting or flexing of the cell. This is pretty common for like spirochetes. It's what gives them that like helical shape, that spiral shape, that telephone cord shape. For attachment and mating, we talked about the fimbriae and the pili, right? So the fimbriae really just for attachment, and then the pili for transmitting like genetic information. Okay. Flagella sometimes can be used for attachment, but I really want you to think of flagella for movement and the pilus and the fimbriae for attachment. Well, the fimbriae for attachment and the pili for the sex. It's not for sex, but you know, it's similar. It's as close as you're going to get with these. So they exchange like DNA, usually typically plasmids through this way. So moving on. The sex pilus, it's long, it's rigid, it's tubular, it's made of pilin protein. Only gram-negative bacteria are going to have it, and it's used in conjugation. So exchange of DNA, basically. And it is controlled genetically, not all, just make it, it has to be something that is triggered. So the GLYCOCALYX is a slime layer. The GLYCOCALYX, so when I think of GLYCOCALYX, I think of the slime layer, but it does include the capsule as well. It's a tighter version of it. But anyways, the whole point is the GLYCOCALYX, the glyco is for sugar and calyx is like coating. That's what that word means. It's the sugar coating. So the slime layer is loose and it protects the cell from loss of water and nutrients. The capsule is tighter, like I said. It's thinner and it's denser and it's a lot stickier, and then things can't bind onto it like antibodies and stuff. Okay. Can't eat the cell up as well. Whenever our body is trying to go eat up bad guys, it can't grab onto them as well. So we have this thing called a capsule stain. You'll be doing them in the lab. This is the capsule stain. The capsule is the white, the reddish on the inside is the cell, and the outside is just the negative stain for the background stain. So you can see here, like you see organisms over here on the left versus the organism on the right. You see that slimy ickiness. That slimy ickiness is from that GLYCOCALYX. So what functions can the GLYCOCALYX have? Well, we talked about the capsules. They're formed by pathogenic bacteria and protect against phagocytic white blood cells. What's a phagocytic white blood cell? Phagocyte, phago means eating. A phagocyte, cyte is like cell, okay? So phagocyte is an eating cell. What does it eat? It eats bad guys. It eats pathogens. A pathogen is a microorganism that causes disease. Yeah. So it'll eat anything that's foreign, anything that it sees as not from us. Okay. Biofilm. Biofilms, examples of these are like the plaque on your teeth and also long-term indwelling artificial devices. Apparently, I don't think much about those, but things that are stuck in the body for a long period of time can grow their own little bacterial films on them. So they can communicate with one another within that biofilm. Essentially, what you get is you get a bacteria that come along, they stick onto a surface. They get their little coating with their little GLYCOCALYX. They stick up onto one another. They keep multiplying. Other things get invited into the system, and they talk to one another and make sure we're all living all good and keep on living and growing and so on and so forth, as long as they're getting information from one another saying that life is good. All right, moving on to cell envelope. The cell envelope is composed of 2 or 3 basic layers. The cell wall, the cell membrane, and sometimes the outer membrane. Okay, just definitely at least the cell membrane. Together, these things, the cell wall, the membrane, and the outer membrane, they create a protective unit that we call the cell envelope. Okay. The gram stain. This is what we use to determine if something has the presence of the thick cell wall and only just the one membrane and the thick cell wall, or there can be a one membrane, thin cell wall, and another membrane. Okay. And then we have terms for those. Those are gram positive and gram negative, respectively. And I'll show you in a moment what I'm talking about. But gram stain was developed by this guy, Graham, and it allows us to tell the difference of the two based on their cell envelope, but basically their cell wall. I want you to focus on that the most here. Okay. You have to know this for sure. So this is gram positive versus gram negative. Gram positive, we got this thicko cell wall going on with this purple structure here, and then the cell membrane is a normal thing. On the gram negative, we've got a membrane, normal membrane, a thin little cell wall, and another normal kind of membrane. Okay, so that's the difference as far as the structure of the outside of gram positive versus gram negative. What makes a gram positive positive and what makes a gram negative negative? The cell wall, the thickness of it. You see how thick that sucker is on the gram positive? That is going to trap more dye. That's what this is about. That's what this is all about. Okay, on the gram stain side. They also have this stuff in it called teichoic acid and lipoteichoic acid. If you see anything teichoic acid, then you know it's gram positive. That's helping keep all of that structure like a scaffolding all stuck together. Okay. I don't know if I want to talk about the gram stain right now, but let's just go through it really quick, I guess. The gram stain, basically what happens is you have that crystal violet first. Crystal violet gets in there and gets all stuck up in that thick cell wall of the gram positive. Then we add iodine onto it. The iodine is a mordant. I'll think of like brick mortar. Think of that. It's a mordant. It makes the crystal violet stick. So if the crystal violet is going to get stuck up in there and stay, the iodine makes it stay. Okay. And then we rinse with alcohol. So that rinses out all of the unbound crystal violet. Well, it didn't bind in the gram negatives because the gram negatives don't have enough cell wall to trap it. It's just not enough. So it gets rinsed out. So if you were to look at it right then, you would look at it and see that you have purple gram positives and then clear gram negatives at that point, if you did it correctly. Then we counter stain with safranin. Safranin is just a red stain, pinkish stain. And then that allows us to see the gram negatives if they got washed out properly, you know, like they're supposed to. It's not supposed to have any purple in it. So that's why it works the way that it does. And the purple is real dark, so you just don't see it quite as well as the purple is real dark, so you just don't see the safranin quite as well because of how dark the purple is from the gram stain, the crystal violet. Okay, right. Gram positives have the thick cell wall. Their cell wall is made of peptidoglycan. They have also an inner cytoplasmic membrane. The gram negatives, however, have an outer membrane, a thin cell wall in the middle, and then an inner membrane. So there's 3 layers there, but that gram positive had that thick peptidoglycan layer. Okay. So what's up with the cell wall? What's it do? It helps us determine the shape of the bacteria and provides strong structural support, keeps us from bursting in the presence of osmotic pressure. Osmotic pressure is like the difference of water versus salt concentrations in and out of the cell. Peptidoglycan, right? So most bacteria have peptidoglycan in their cell wall. We have peptidoglycan in the gram positives and the gram negatives, but the gram positives just have a lot thicker of it. And that's the difference. So so most bacteria do have peptidoglycan, and it provides a strong but flexible framework. Peptidoglycan, what is it? It is, we have a disaccharide, and you don't have to know necessarily these exact words that are on here. You might want to, like, if I said them out to you, be aware of what they were, but like peptidoglycan basically has a disaccharide, NAG and NAM, N-acetylglucosamine and N-acetylmuramic acid. So glucosamine versus muramic. Okay. That's the repeating units of that. Just over and over and over and over again. Okay, those two. So we got repeating units of NAG and NAM. And in the gram-positive, we are linking those across from one another using those teichoic acid peptides. Okay, so we have peptides linking across and peptides coming back up to link to the next layer and so on and so forth. Okay. I don't know why that's 2 slides. Structure continued. So, I mean, I don't know what else to say about this, but they're linked by these bridges. And then the teichoic acid comes and will stabilize all of this across like the whole way. Okay. So it's just like a scaffolding, how it's set up with the peptide cross linkages. And then the teichoic acid is going to just stabilize everything. So yeah, lipoteichoic acid going up, and then teichoic acid over here as well. So that's just going to, like I said, stabilize that whole structure. That's the gram-positive. It's thick. It's a sheath of peptidoglycan that is about 20 to 80 nanometers thick. Right, like I said, the teichoic and lipoteichoic acid are involved in stabilizing the cell wall. That's part of the cell wall maintenance, just maintaining the structure as it is around the whole cell. It'll also help with the cell when it is enlarging the cell wall for cell division. And that has an acidic charge on its surface. So that gives it an acidic. All right. So moving on to the acid-fast bacteria. So this is part of gram-positive. If you were going to have a gram-positive bacteria that was like slightly changed and off from like what is expected, that would be the acid-fast bacteria. This includes Mycobacterium and Nocardia. Yes, you have to know that. This is absolutely another thing that will be tested on. Mycobacterium, you're going to know this for a long time. This is going to be on a lot of tests. And on the lab tests as well, definitely recognize Mycobacterium. If you can't know Mycobacterium and acid fast, then you're going to have a problem. Acid fast, think Mycobacterium. Mycobacterium, think acid fast. And what was the other one that I said was similar to that? That was the endospores and the bacillus and clostridium, right? You see bacillus or clostridium, you think endospores. You see endospores, you think clostridium and bacillus. So same thing with the Mycobacterium. Mycobacterium, you think acid fast. That should just instantly pop into your head and be in there. Right, so they have something called mycolic acid. That's why they are called Mycobacterium. So they have this acid in there that's sort of like a wax that helps increase the structure, basically, of the cell wall. So it makes it a little bit stiffer, a little bit more hardy. So that's like tuberculosis and leprosy have these. Right, the negatives. They have a very thin peptidoglycan. They have a somewhat rigid structure thanks to like the two membranes kind of supporting that peptidoglycan structure, but that's pretty much all it has going for it. They have also special polysaccharides called lipopolysaccharide in the membranes, the outside membrane, lipopolysaccharide and endotoxin. So it's part of the lipopolysaccharide features. Endotoxin is something that is part of gram-negative. And this is going to be asked a lot as well throughout the whole of the course. I'll ask some questions on exams about endotoxin, because this is a repeating thing. Like we're going to see it a lot throughout the entire course in all the different units about endotoxin and where it comes from. If you see endotoxin, like we've already got what's going on with acid fast, we've got mycobacteria. We've already got with the endospores, we've got bacillus and we've got clostridium. Now we've got endotoxin. If you hear endotoxin, you need to think gram-negative. Okay, so gram-negative endotoxin. Okay, gram-negatives have endotoxin. Gram-positives do not have endotoxin. If you see endotoxin, that is for gram-negative. I'm not going to confuse you with the stories about the gram-positive stuff. So I just want you to focus on that for now. That's all I care about with this. Lipopolysaccharide, that's the outer layer of the outer membrane, the outermost. We have three basically parts of it. The lipid A, which is the actual endotoxin, the core, and the O antigen. The O antigen is probably the dominant antigen of gram-negative cells. You can't make a vaccine against endotoxin, because like we have so many bacteria in our gut, and I mean a lot of bacteria in our gut. We don't want our body fighting those off. We need those to survive. So you don't want to have that mess going on. This is going over the gram stain procedure. Now that we've talked about gram positive and gram negative, remember, gram positive, we'll add crystal violet, it turns purple. We fix it in place with the iodine, we rinse it, it doesn't come out, and we dye it with safranin, which is red. You won't see it because the purple is so purple. Okay, gram negative, you put the crystal violet, you add the iodine. At this point, it is still going to be purple because you haven't rinsed it out yet, right? But it's not going to get fixed in there by the iodine. So now you're just going to rinse it out with the alcohol and it'll be clear. And then you counterstain with the safranin, which is the red dye. So if you isolated a bacterium from your skin and chemical analysis shows that it contains proteins, peptidoglycan, lipids, DNA, and teichoic acid, what sort of bacterium would this be? Remember, teichoic acid, whether it's lipoteichoic acid or just regular teichoic acid, that's a gram positive cell wall thing. Cool. All right, mycoplasma. So mycoplasma lack a cell wall. I mean, if you see mycoplasma, just think no cell wall. They just got the membranes, okay? So they are stabilized with sterols, and they're a little bit more resistant to lysis because they're more squishy, essentially. They can have a little more give. They have a pleomorphic shape because of this. The cell wall often imparts the shape of the bacteria because they don't have a cell wall. They can just be kind of whatever shape they need to be. The cell membrane itself. So this is the sheet that's molded around the cytoplasm, the inside cell membrane. The lipid bilayer. Remember, we have like the heads on the outside and the tails on the inside that are hydrophobic. So we have phospholipids and proteins scattered throughout. And see how it's mostly proteins. That's neat to think about because it's mostly channels that are going to be controlled to allow things to come in and out, as well as other proteins that are going to help with metabolism and things like that. So mycoplasma have a high amount of sterol within all of this. That's their difference. And ARCHAEA have hydrocarbons rather than fatty acids. So what's going on in the cell membrane? What does it do? Well, it contains everything, obviously, but like it's important in energy reactions. A lot of that goes on within that inner cell membrane. It's involved in nutrient processing as well. Remember, they don't have organelles that are going to take care of this. They don't have parts inside of them that take care of this. They have to do all of it within just the junk of their cell. So synthesis of anything is going to be going on a lot of times on the inside of that membrane, and then transport, of course, in and out of the cell. Membranes are important as far as when we're talking about like protecting what's inside and keeping what's in and keeping what's out. Part of that involves selective permeability. This is a term to be familiar with. Selective permeability refers to water and other small, very small, uncharged molecules that can diffuse freely across the membrane through usually different channels. So special carrier mechanisms will exist for the passage of most other molecules. Okay, so there's some other way to get other things across, but only water and very, very other small things can diffuse on their own. Usually, typically only water, and that's the one I want you to focus on. Water can diffuse freely, pretty much nothing else can. They need a way to get across the membrane. Secretion is a term that refers to discharge of metabolic products into the extracellular environment. That's just a term to be aware of. That's something that needs to happen with regards to the membrane itself. Cytoplasm, what's going on in the cytoplasm inside of the cell? The cytoplasm is a gelatinous solution that is contained by the cell membrane. It's a prominent site for cells' biochemical and enzymatic activities. The cytoplasm itself is 70 to 80% water. It's a complex mixture of sugars, amino acids, salts, and contains chromatin, ribosomes, granules, and fibers that act as the cytoskeleton. So basically a lot of support going on there, and just a lot of enzymatic activities, and like just everything else that's going on that wasn't going on the cell membrane is going on in the cytoplasm. It's pretty much, they don't have, like I said, they don't have places for stuff to be going on. So it goes on inside anywhere. The chromosome of the bacteria. Bacteria have a single strand of DNA as their chromosome, and it is a circle. And that's it. They don't have this nonsense that we have. Okay, they don't have this. No, they don't have that. They have a circle 1. That's their chromosome. Their normal genetic makeup. Their normal genetic makeup is just 1 circular strand of DNA. We contain it in a region of the cell that we refer to as the nucleoid. Like I said, they don't have any way to like wall it off or anything. So it's just in a general area called the nucleoid. It's tightly coiled around proteins, so it can fit into that kind of compartment, that area of the cell. Then we also have plasmids that can be involved. They're also circular, but they're a lot smaller, and they just contain extra genes that are going to help that bacteria survive a little bit better. So they're non-essential. They don't need them to live. They are separate, double-stranded DNA, just like the regular DNA is double-stranded DNA. 2 strands of DNA, they go back. One reads like in one way, and the other's the match of it in the other way. It's duplicated and passed on to offspring during replication, just like the regular DNA would be, but it's not considered part of the genome. It's not considered part of the chromosome because it's not part of the chromosome. It's on its own. You can lose it as well. It also confers protective traits and is important in genetic engineering, which we will be talking about later on. We call it transformation. Ribosomes. They have ribosomes. Ribosomes are made of RNA and protein, mostly RNA. They have a bit of protein to them. That's called ribosomal RNA. That's what makes up ribosomes, ribosomal RNA and protein. You find them all throughout the cytoplasm. They're super tiny. They make protein. That's what they do. They read the messenger RNA, which is that little bit of the gene that's telling you to make a certain protein. They read that and then spit out protein. Okay. What's protein made of? Do you guys remember what the subunits of protein what subunits are? It's proteins are made up of amino acids. So they're going to literally the ribosomes will take amino acids and link them together based on what the mRNA, that messenger RNA from the gene, is telling them to put. So that's how that's going to work. We'll talk about it more in a later chapter. All right, ribosomes, we measure them as far as their size goes in Svedberg units. I am not as worried about that. You know what this is, what they centrifuge it. Basically, that's like high speed spinning. And based on how they settle, determines their size. And they just attributed these numbers to them based on that. Okay, so bacterial ribosomes are 70S and eukaryotic ribosomes are 80S. Then we got inclusion bodies where you store things. Here's some pictures of inclusion bodies, big and small. All right, the cytoskeleton polymer is a protein similar to like what we have actin in eukaryotic cells. You guys are probably not familiar with that, but that's okay. I'm telling you that eukaryotes like you and I, our cells are shaped by interior structure made by actin, okay, actin filaments. So they have polymers of proteins similar to that in prokaryotes, like bacteria and archaea. That helps with their cell shape. So moving on to endospores, which we mentioned previously. Endospores. Which ones make the endospores? Did you already forget? I'm so, I don't know, disappointed, I guess. I don't know, because maybe you remembered. I have no idea. Endospores, Bacillus and Clostridium. Okay, so endospores. They withstand hostile conditions and facilitate survival. So in a hostile condition, you don't have any water, got way too hot outside, it's too dry, you don't have any food available or whatever. There might be radiation, whatever. There's a lot of them. Oh, it's on the next slide, apparently. Whatever. Endospores, they keep you alive. They are not metabolically active. They are alive. They are not metabolically active. They are inert, but alive, resting, but alive, and lying in wait, waiting for the good stuff to come back. The vegetative cells, that's the normal ones that are metabolically active, that are normal, doing the normal stuff, dividing and all of that stuff. And sporulation is a term that refers to spore formation, which is making the endospores. If you are a vegetative cell and you're like, oh man, stuff's getting real, I need to hunker down. That act of hunkering down and making your endospores That's sporulation. Sporulation is making the spores, the endospores, okay? Endospores, they can resist heat, they can resist drying, they can resist freezing, radiation, and chemicals. That's a lot. It's pretty impressive. What sort of things might, stimulate you to make an endospore? You might not have nutrients, carbon and nitrogen sources going low. That's a pretty big one to trigger something to go into making endospores. So the sporangium, because we're just going to jump around in all these terms because why not? The sporangium is the term referring to the sporulating cell. So you're a vegetative cell, things are fine. They start running out of food. There's like absolutely, you're having a real hard time finding like a carbon source. And you're like, oh geez, I should probably sporulate. So you start going through sporulation. What does that mean? Making endospores. Okay. Now that you are doing that and you're starting to get ready to make your endospore, you are the sporangium, and then you've got the endospore forming in you during the sporulation process. Okay, here's some pictures of it. Vegetative cell, not real happy, decides it is going to do an endospore. So now it is a sporangium. Okay, so this is sporangium. And then this is the forespore. That's the spore, not 4 and 7 years ago. That's a forespore. It's at the one end. The sporangium, it starts to engulf where it's making the spore. It has a little bit of spore stuck into it once it's engulfed it. The sporangium now is making even more of it into a spore. The sporangium has now made it a mature endospore that's just waiting. So now we are releasing the endospore. The sporangium is going to die and go off probably and not have to exist anymore. But the endospore will go and lie in wait. Still alive, right? But it's lying in wait, waiting for good conditions. Good conditions come up. It can turn everything that's inside of this whole cortex, that coating on the outside. It can break out of that and just turn back into a vegetative cell. I don't know why this never works. This. I don't know. I tried to set it up so that you could watch this, and I thought that maybe I can't. I guess I just imagine that these things are going to work. I'm so sorry. They work in the classroom. I don't know why. Oh, here we go. Okay. Sorry.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into 2 complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by 2 membrane layers. Next, peptidoglycan is laid down between the 2 membranes of the developing spore to form the cortex. Dipicolinic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed.
Don't worry about the details of this.
A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell and the mature spore is released.
Okay. Moving on. Now we figured that out. Did you just put the pen away? Apparently, my bad. Okay, so germination begins when favorable conditions arise. Exposure to water and germination, and a germination agent, whatever that is, whatever triggering thing that thing needs, whether it's the carbon source or a nitrogen source or whatever it is, it gets exposed to that and some water, and it can start germinating. So that means going back to the vegetative form. So germination will start with hydrolytic enzymes breaking down the cortex, just breaking down that outer coating, rehydrating the core, taking up nutrients, and bacteria growing out of the coat. And then once that happens, this is usually about only 1.5 hours for it to go from that endospore into a vegetative cell again. Like reanimation. All right, so why do we care about endospores? I know like you guys don't necessarily give a crap about like bacteria that I might be talking about that like live in the soil and whatever, because like you're probably going on into healthcare field or dental or vet, which are pretty much the same. I mean, I need to know the similar amount of information. So what do you care about an endospore? Well, there's a lot of diseases that are existing out there because of endospores, essentially, that are dangerous because of the endospores. That includes anthrax caused by Bacillus anthracis, tetanus caused by Clostridium tetani, C. diff, Clostridium difficile, that's pseudomembranous colitis. If you have ever worked or even heard of hospitals, pretty much, then you know what C. diff is. C. perfringens, that causes gas gangrene, which is whenever like gas will build up inside the tissue, you can lose whole limbs as a result of that. And then Clostridium botulinum, which causes botulism, or, you know, makes Botox, however you want to look at that. So yeah, so you notice these are all Clostridium, and this is a bacillus. But yeah, bacillus and Clostridium, that's your endospore, guys. All right, Clostridium and bacillus, right here. So endospores are constant intruders where sterility and cleanliness are important. It's hard to get rid of them. It's really hard to get rid of them. They resist the ordinary cleaning methods. They frequently contaminate cultures and media. Hospitals have to protect against them in wounds. You know, destroying them in the food canning industry can be difficult as well. So that's something you have to, you know, always be on the lookout for in any of these industries, trying to get rid of endospores. So if we are making endospores, that's sporulation. Remember, sporulation, we are making spores. Okay, archaea. And archaea are probably more closely related to eukarya than they are to bacteria. That's just genetically speaking, okay? They have a similar RNA, the ribosomal RNA sequences. They don't share that with bacteria at all. They're very genetically distinct as far as that goes. Their protein synthesis and ribosomal subunit, the parts of the ribosomes, they're very similar to eukaryotes as well. They're probably the most primitive of all life forms. They're closely related to cells from 4 billion years ago. Live in habitats similar to extremes back in the olden days: heat, salt, acid, pH, pressure, atmosphere changes, and stuff like that. They often can be methane producers. Hyperthermophiles, they like really hot temperatures, actually have to have hot temperatures. Extreme halophiles can live in very high salt concentrations, and sulfur reducers. They live in these extreme environments, okay? Methanogens, these are the ones that convert just carbon dioxide and hydrogen in the atmosphere into methane gas. They live in anaerobic swamps. They don't get any oxygen there, in the mud, and the bottom sediments of lakes and oceans, and the digestive systems of animals, which we are probably aware of from the smell. You know what methane smells like. Gas produced in the swamps can become a source. We may be able to use that gas made in the swamps as a source of fuel, but it also does contribute to global warming. It's a greenhouse gas, so it's a pretty significant source of it. The halophiles, the extreme halophiles, the ones that need the salt, they like high salt, but they also have to have salt. They require salt to grow. Halo, salt. Why halo? When you see halophiles, don't think of halo like an angel. No. Halo, like a halogen bulb. Yes, like that. Like if you guys took chemistry and you know the halogens, that's what I'm talking about, like chlorine, right? So that's what we're talking about here. The halophiles, they like that. Think of salt when you see it. Halophiles, they can multiply in 36% sodium chloride. That would destroy almost every other kind of cell out there. They exist in inland seas, salt lakes, salt mines, and in salted fish. Notice the word salt here. They use red pigments to synthesize ATP. That's their energy molecule, remember? Adenosine triphosphate, we lose a phosphate, we get a ton of energy, we can power the things we're doing in our cells. And then they do that in the presence of light with that red pigment. So here's a wide expanse of a bunch of archaea growing out there, getting some sun and making their energy. We also have these psychrophiles. The psychrophiles are the archaea that can grow at the very low temperatures, the very cold temperatures, versus the hyperthermophiles. And do think of thermo as being like temperature. So thermophiles, they love temperature of high temperature. The hyperthermophiles flourish at high temperatures. Now, what's a high temperature? What am I talking about here? They flourish between 80 and 113 degrees Celsius. They cannot grow below 50 degrees Celsius. So what do these numbers mean? Do you guys know what temperature water boils at? Water boils at 212 degrees Fahrenheit. That's 100 degrees Celsius. They flourish at boiling temperatures. So that's nuts. That's nuts. They're also often, these guys that like those high temps, they also like the salt and acid a lot of times. Okay, so archaea. What type of organism is an archaea? What kind of a cell does it have essentially? So they are not eukaryotes. Those are the ones that have true nucleus. They're not viruses. Viruses are not alive. Remember? Fungi are eukaryotes, so they would have to be prokaryotes. All right, moving on to Bergey's manual. Bergey's manual is the definitive published source for bacterial and archaea classification. You need to know what kind of bacteria or whatever that you're dealing with. You will look it up, whatever feature you know about it in Bergey's manual, and then you can use like deductive reasoning to try to figure it out from there. Bergey's manual of determinative bacteriology uses phenotypic characteristics. So that means stuff that you can see of it, not genetic. That's the point there, not genetic. So maybe their tests of whatever kind of media they can grow on and stuff like that, or what their colonies look like, or what the cells look like, or how the cells group together, stuff like that. Okay. Cell shape, how they behave in culture, and their biochemical reactions. That's what I was just talking about, right? So we still use it today to quickly identify unknown bacteria, but usually the online version. The systematic bacteriology combines all of that info with ribosomal RNA sequencing for classification. So you can use actual genetic information for classifying with that. So determinative uses just phenotypic. Maybe that's all you have available. So that's what you would use. If you have your ribosomal RNA information, then you would use the systematic version. Moving on to taxonomy of these bacteria and archaea. We have 4 groups to be aware of: the gracilocutes, the firmicutes, the tenericutes, and the mendozocutes. Okay, so gracilocutes. So here's how I will break this down, and it might not be helpful for you, but I hope that it is. Firmicutes. If it is firmicutes, these cute guys, these firm cute guys, they are firm because they have strong cell walls because they are gram-positive. Remember, gram-positives, because they stain positive with that purple dye and the gram stain, that means that their cell walls are really thick. It traps the stain, right? So they have thick, strong cell walls, which are nice and firm, firmest of all of these, and we will call them the firm acutes. That's the gram-positives. The gram-negatives, I just remember it because it's gra, so that's like gram. So that's the next one with gram. I don't know. That's probably not super helpful, but there is that. The other ones, mendozocutes. Mendozocutes, I dated this guy in, I guess, middle school or high school. I don't really remember. And his name was Joey Mendoza. And he was like the oldest, like first boyfriend I ever had or something stupid. And so I just think archaic, because that's archaic, because I'm old now and very much less naive. But anyways, Joey Mendoza, that's who I think of when I see Mendoza cutes. And so maybe you guys will think of that too. Mendoza, it's old. That's from the olden days. ARCHAEA, archaic, right? Okay, and then that just leaves tenericutes. So tenericutes, I don't have a good one for that one yet. They lack a cell wall. So these are the soft ones. So these are like the mycoplasma. All right, diagnosing divisions in the diagnostic scheme. You have the gram positives and the gram negatives and bacteria that do not have cell walls, like, you know, the mycoplasma. So that's how you would group them at first. So gram positive, you can also arrange that, shoot off into the acid fast because they stay in gram positive. Subgroups that are involved, we have based on cell shape, remember cocci, bacilli, spirillum. Then we have the arrangements, so staphylo, strepto, palisades, and stuff like that. And then we have oxygen usage. Does it grow in oxygen? If it does, if it needs oxygen to live, it's aerobic. It has to have oxygen. Aerobic, okay? If it's anaerobic, think of an as being like no aerobic, no air. Does not use oxygen, no oxygen. And if it's facultative, just think of it, it can facilitate both, facultative. But it can survive without oxygen. It can also use oxygen. We'll just write that on here so that you guys can watch my scripturally handwriting. So that's the idea with how you might break these down and start looking at them, maybe in Bergey's manual, or start separating them out in however you want to separate them out as scientists. So to summarize what we've gone over in chapter 4, the prokaryotes, we talked about the difference between prokaryotes and eukaryotes. We compared and contrasted them in a bunch of different features. We talked about the bacterial appendages, the flagella for movement, the fimbriae for the adhesion, the pili for exchanging DNA, capsules and slime layers, the sugar coatings that help with sticking onto things and protection. Shapes of cells, the cocci, the bacilli, the spirillum, and the arrangements of cells, the staphylo, the strepto, the palisades, and whatever. Then we have the cell envelope, which includes the membrane, the inner membrane, the cell wall, and the outer membrane if it has one, right? So we talked about peptidoglycan and how it's thicker in the positives and the negatives and how the gram stain works. We talked about other deviants like the acid fast and the mycoplasma that don't have a cell wall. Then we have internal structures. We discussed quite a bit about the endospores. We talked about archaea and where they live in the extremes. And then talked about a little bit about the taxonomy. I feel like we didn't touch too much on that, the firmicutes and all of that. So yeah, that's that for chapter 4. If you have any questions about it, contact me and let me know. And I can't wait to move on to chapter 5, I guess. What is chapter 5, you guys? Chapter 5 is eukaryotes. I can't wait till unit 2. I can't wait. So I'll see you guys next time. All right. All right. Let me take forever stopping this video really quick and see if we can.